A las personas que todavía no se han ubicado, por favor, les pedimos que tomen asiento.
Please take your seats.
Participants who still are on the part of the back, please go to your seat. We're going to start right now. Muy bien, buenas tardes a todos y todas.
Good afternoon, everybody.
Ya vamos a empezar la segunda parte del evento.
We are going to start this second part
Les
of
doy
the
la
event.
bienvenida nuevamente a la
I welcome you
Conferencia
again to
Internacional sobre
the
Carbono Azul y Humedales
International Conference on Blue Carbon and
2025
Wetlands
y
2025.
una bienvenida especial a
And
quienes se unen a
we extend
nosotros,
a
ya sea
special
aquí
welcome to
en
those
físico en
joining
Santa
us for the
Marta
first
o a
time
quienes se unen de
here in
manera
person in Santa
virtual.
Marta, or those that are
Antes de comenzar,
joining
vamos a dar
us
unos pequeños recordatorios.
online.
Recuerden que las personas que
Before we begin,
deseen
remember
ir a la
that
salida de campo el
those
día
that are
jueves
interested in coming to
deben
our field visit
acercarse
on
al
Thursday,
área de
please
registro que se
come to
ubica
the
en la parte del
registration
fondo,
desk
en la parte
found
del
in
lobby.
the back
Entonces, por
of the
favor,
room outside in the lobby.
acercarse para
Please
inscribirse. Solo son
go
50
there and
cupos.
enroll your
Adicionalmente,
names.
recuerden que
In addition to that,
hay interpretación mixta
there
en
is
inglés y en español para los formatos
interpretation
de
in
las
English and Spanish
presentaciones.
for
Entonces, si aún no
our
tienen
presentation. So if you
su
still
audífono,
haven't
se pueden
collected
acercar a
your
la parte de atrás
headset,
a
please
conseguirlo. También tenemos la aplicación
go
Juba,
get
como les
yours. We also
mencioné en la
have a,
parte de la mañana. A través de esa
Hoover
aplicación pueden
application,
enviar
and
sus preguntas en
through the
caso de
application you can post
tener
your
alguna
questions in
pregunta.
case you have any.
Si están publicando sus
If you're
fotografías a través
posting
de
on
LinkedIn, nos pueden
LinkedIn
etiquetar
during the
como
conference, remember to tag
@bluecarbonmrv.
us as Blue Carbon MRV.
Este próximo bloque
This
técnico se enfoca
next
en las
technical
innovaciones
block
científicas o en
focuses
las
on scientific
investigaciones sobre
innovations
carbono azul.
in blue carbon
La
research.
ciencia es la base para
Science
la
is the
acción
foundation
de
for
carbono
credible
azul
blue
creíble.
carbon
En este
action.
bloque
In this track,
exploraremos
we
herramientas y técnicas
explore
de vanguardia.
cutting-edge tools and techniques
En ese sentido, vamos a explorar el análisis impulsado
to
por
explore
inteligencia
the
artificial, es decir,
analysis
la
driven by
redefiniendo cómo
AI that's
medimos,
redefining how
monitoreamos
we
y modelamos
measure,
los
monitor,
ecosistemas
and
de carbono
model
azul.
the blue carbon
Nuestros
ecosystems.
ponentes
Our
compartirán
speakers will
innovaciones
share
que no solo
innovations
mejoran la
that not
precisión, sino
only improve
también
accuracy
amplían
but
lo
also
que es
expand
posible
what
en
is possible
restauración,
in
gestión
restoration,
y alineación
management,
con
and
políticas
alignment with
públicas.
public policies.
Nuestro primer
Our first
ponente es el
speaker is
doctor Tobias
Dr.
Jackson,
Tobias Jackson,
investigador asociado senior de
Senior
la
Research
Universidad de Bristol.
Associate at the
Dr.
University of Bristol.
Jackson es un
Dr. Jackson is
científico
a
costero cuya
coastal scientist
investigación
whose
combina
research
ecología
combines
de campo, mapeo
field
avanzado y
ecology,
análisis
advanced mapping, and
geoespacial para
geospatial
mejorar
analysis to
nuestra
improve
comprensión
our
de las dinámicas de
understanding of
carbono en
carbon
los
dynamics
bosques de
in
mangrove.
mangrove
Su
forests.
trabajo integra
His work
datos
integrates
terrestres y
terrestrial and
aéreos con
aerial
mediciones en situ
data
para
with in
producir
situ
estimaciones
measurements to produce
precisas de
accurate
reservas de
estimates of
carbono
carbon
que puedan
stocks that can
guiar la
guide
planificación de
conservation
conservación y las
planning
políticas
and
climáticas.
climate
Hoy nos
policies. Today,
presenta
he will present
mejorando nuestras
improving our
estimaciones de
estimates of
almacenamiento de
carbon
carbono
storage
en
in
manglares
mangroves
usando
using
LiDAR.
LiDAR.
Le damos la bienvenida
Let's
al
welcome
Dr.
Dr.
Jackson.
Jackson.
I have an old version of my slides, which is my fault because I kept updating it. So I don't actually remember the order of these ones, but I'll try and work it out. So I am going to be talking about allometry. And by allometry, I mean the process of getting from field measurements of height and diameter of trees to the carbon storage or the biomass of those trees. And we're really using the height and diameter to estimate the woody volume of these trees. Come forward. Okay, that's fine. And so we're trying to estimate how much volume, how much biomass there are in these trees. And then we can use that to scale up to the landscape level. But it's only on this narrow allometry I'm focusing today. Oh, that's a shame. Okay, so this is a really nice paper which was published last year. Sorry. So this is a really nice paper published last year where they compiled all of the data out there on which we use for our current allometries. So they found 590 trees which have been cut down and weighed. And this is how we build our allometries. So we have to get a allometric equation. You have to first cut down the trees and weigh them, and then you can use that to build your equations. And this paper found 590 trees in this region, which have been cut down and weighed, and essentially found that they all followed pretty much the same allometry, which was quite a surprising finding to me. But in this analysis, there were only 20 trees larger than 15 meters in height. And so there's very few large trees in here. Okay. Okay. So sorry, things aren't working. To get past this, we want, instead of cutting down trees and weighing them, we want to do this with terrestrial laser scanning. And this is what terrestrial laser scanning data looks like. So this is some data which Jasper collected in Suriname, and it just shows you that there's a nice 3D good detail of all the trees in this plot. And if we just take out a single tree, it hopefully looks like this. You get good detail on a single tree. And this is a really valuable tool because it means we can do the big trees well. So the paper which had all the trees together, they were missing the large trees. Whereas in this tool, the big trees are actually the easiest ones to do because there's more wood per leaf. So the leaves get in the way on the small trees, but the big ones are reasonably easy. And so this is what we do with this. We collect these 3D LiDAR point clouds, and then we fit cylinders to them. So this is a cylinder model of a tree. This is actually a eucalypt tree in Australia, I think. So it's not actually anything to do with mangroves yet. And so we fit cylinder models to them. And you can see the middle picture there is a collection of cylinders. And then if you add up the size of all of those cylinders, that tells you how much biomass there is in the tree. And then in this paper, Kim Calders, who led this paper, cut down the trees and showed that actually this terrestrial LiDAR method, it works really well. So on the x-axis here, you have the trees that he cut down, and on the y-axis, you have the same ones measured using LiDAR. And so now we know that the method works, we can use it to try and improve our allometries. So again, I've got an example here from temperate forests, also by Kim Calders, and essentially they showed that the temperate broadleaf trees that they were studying stored more carbon than was estimated with traditional allometric equations. So on the x-axis here, you have how much biomass there is if you were to use an allometry from the diameter measurements, and on the y-axis, you have if you use the terrestrial laser scanning. And so there was, quite substantially more carbon in this forest than we had realized because of the way we were looking at it. So the LiDAR helped us understand that. In mangroves, it also works. So this is some work that Hamish has been doing. So in mangroves, it's a bit more difficult because the trees branch at the bottom, there's roots at the bottom as well as at the top, and the cylinder fitting expects the trees to only branch outwards. So we have to cut the tree in half. In our point cloud data, the virtual tree in half, and we flip the roots upside down, and we fit cylinder models as if there were two trees, and then we stitch them back together again. And this works quite well in mangroves as well at the moment. So I'm doing some work at the moment to use terrestrial laser scanning data and to collect more terrestrial laser scanning data. Lots of people here are involved in this. And the aim is to basically test whether these allometric equations or whether any allometric equations we currently use work well for large trees. So we're going to put, if you see this graph on the left, there's not very many trees above 10 meters, and with this method, we'll be able to increase the scale of that and see if it still works. These graphs are a bit misleading because they're both in log space, and when you log transform things, everything looks quite straight. But actually, if you were to plot these out in real numbers, it would look quite different. And so actually today, when we get back to the hotel at like 5:30, a few of us are going to have a beer and talk about this project, talk about how we're going to bring together different LiDAR data sets, how more data is going to be collected. And so if anyone wants to join at around 5:30 at the hotel, probably on the rooftop bar, maybe, we have a discussion about this further. Yeah. And terrestrial laser scanning is cool, but it has quite a lot of problems as well. So it's hard to do, right? Especially in a mangrove. So you have to scan from lots of different positions. So what we're planning to do is to target individual trees. So we'll go to existing plots and target some trees and then scan them from multiple angles so that you can capture the 3D structure. And then we want to stitch those different scans together. And that stitching the different scans together requires these targets, these funny white and black things. So you have to put these out in the field and keep them really still while you're scanning. You also have to have good conditions. It can't be windy. So there are problems with collecting the data. But even more than the problems collecting the data, there's lots of problems in processing the data. So this just shows you an example of some data. The top of a tree, which is 25 meters tall of a mangrove, again from Suriname. And it's reasonably good data quality. You can see the main trunk and everything, but there's leaves all over the place. There's also lots of points which could just kind of seem a bit random. Maybe there was a bit of wind or something. And when we just apply our automatic cylinder fitting algorithms, it will make a mistake here. So we need to go in and manually clean this data beforehand. So we need to delete lots of the leaves, tidy up some of the branches. It's quite a slow and process, but it's really valuable if we can get some trees out of this, some good high quality large trees, because for every big tree we can get this way, that's a big tree we don't have to cut down, and cutting down big trees is hard. So that's all I had to say about allometry, but I wanted to just take a step back and think about the other parts of this. So this gets us the biomass, how much wood there is in the forest from the height and diameter measurements we take in the field. If we can improve our allometric equations, we can do that better. But there's also other uncertainties. So I just came across this figure from a paper from 2016. I don't know if anyone's seen this, of essentially showing that if you sample wood cores up the tree, it gets less dense as you go. And so this, if we usually sample wood at 1 meter height around, and that would, if we apply this would suggest that there's actually less biomass we thought. And I imagine that there's very few areas with these data. And so I guess that would add quite a lot of uncertainty. So it's just an example of where there might be some uncertainty coming from. And so this is just my best guess of where maybe some of the uncertainties are. So the y-axis is how much these different factors contribute to the overall carbon storage in these forests. So the allometry one, this is what I'm mostly working on. And my job is really to improve our certainty on this, is to make those little white bars a bit narrower. That's my aim. And I think it's an important subject to do. But as I just showed, wood density also introduces some uncertainties. And then there must be some uncertainties in the below ground roots and the dead wood storage. And then soils, I've put as really high. This is just my guess, but it obviously store a lot of carbon, and there's lots of uncertainty there. And I would like to better understand how we might constrain that uncertainty, how big that uncertainty is at the moment, and how we might better constrain it in the future. So this is something which I don't know the answer to, and I think there's probably some people in this room who know the answer to. So over the rest today and tomorrow, maybe we can have a chat about this. Yeah. I actually completely changed these slides on the plane on the way here. So this wasn't the slideshow I was intending to give. So apologies for that. But yeah, I hope I've made my point. Thank you.
Bueno, muchas gracias al señor
Okay, thank you very much to Dr. Tobias Jackson.
Ahora escucharemos a la doctora Noelia
Now we're
Jimenez
going to listen to
Martínez,
Dra.
jefa de
Noelia Jimenez
perspectivas
Martínez,
e inteligencia
Chief of
artificial
Perspective
en
in Artificial Intelligence and Nature Metrics in the United
Bienvenida.
Kingdom.
La doctora Jimenez
Dr.
lidera la
Jimenez is
integración
in
del
charge of the
monitoreo de
integration and monitoring
biodiversidad
from
con
biodiversity
inteligencia
with artificial intelligence
en Nature Metrics,
in Nature Metrics
ayudando a
and
gobiernos,
helping
empresas y
governments, companies,
ONGs
and
a medir el
NGOs to
cambio
measure
ambiental
the
a gran
environmental
escala.
change on a bigger
Con
scale.
experiencia en
With experience in
y
bioinformatics and genomics,
la conservación se especializa
she's
la
specialized
aplicación
in
de
conservation and the application
ADN
of
ambiental
eDNA
e
and
inteligencia artificial
artificial
para
intelligence
evaluar
to be able to
los
evaluate
ecosistemas
the
y indicadores
ecosystems and the
climáticos de
key climate
biodiversidad.
indicators for biodiversity.
Su presentación se
The
titula
presentation is
Uso del
called
ADN ambiental
eDNA for
e
Blue
inteligencia
Carbon,
artificial
Artificial
en
Intelligence
MRB
and
y
EMRB
contabilidad de carbono.
and carbon accountability.
Le damos la bienvenida a la
Okay, so
doctora
we're now
Jiménez, por
giving
favor.
a warm welcome to Dr. Jimenez. Esta no es la presentación, es así.
Hola, ¿qué tal? Thank you very much for the invitation. I'm very pleased to be here. I am bilingual because I am originally Argentinian, but I'm also British. I'll conduct the talk in English because I prepare the slides in English, but feel free to interrupt me in Spanish if you want. So my presentation today, it's about how can we use eDNA, and I'll introduce that concept for the people that are not familiar. And what do we do with AI and the latest disruptions in this space to track and understand better the MRV systems and how this can be aided by the new technologies. My title there is not the same as in the program because in the latest months, I was promoted as a Director of Innovation and Scaling at Nature Strategy. But yeah, so the company is Nature Metrics, is a UK company, and it's specialized in performing different eDNA sampling, amplifying the DNA in the lab, and producing insights that help decision makers in different aspects to improve, restore, and basically monitor biodiversity. So thank you so much for the opportunity to talk about this. So for the non-familiar people, eDNA is basically environmental DNA. So DNA is the genetic materials that every living organism has. And these are cells basically, shared by different individuals. And this is through the feathers or the feces, mucus, pollen, think about that. So this is distributed everywhere. And there are ways to collect the DNA. The company has specialized in making this accessible and cost-effective. So we can now put back this DNA into the lab, amplify it, and understand what species do we have there. So one of the interesting things of DNA is that you can go on a very high level or in a very detailed level sometimes and understand the species level. Some of the problems we've been discussing today are, okay, I have this forest and I need to understand the species in this forest, or I need to understand, for example, what Tobias was describing, how much carbon they trap, but we don't know, we cannot really go to the ground sometimes and measure what species are there. This is the way to go to the ground. This is what we call the ground truth, which is tracking DNA. So the methodology of DNA is basically, we decide we want to understand what species are present in a certain space, and think about this could be applicable, and I will talk about this for sediments and mangroves and seagrass. You do a sampling strategy with the ecologist of the company. So we tell you where exactly to put the samples. We send you these samples. The samples are very easy to use, and this is where it becomes fun because you can involve local communities. Local communities can sample very easily and just send this back to the UK, where the labs will amplify and discriminate among these different taxonomic groups. For blue carbon is in particular eukaryotes, that it will be the assay that we need. Is this a pointer? Yes, great. So we can identify vertebrates, fish, mammals, freshwater insects, invertebrates, bivalves, the eukaryotes, bacteria, and fungi. And then, well, when you have all this, the pretty thing is that we can also understand how they are informing about the health, different functions of an ecosystem. And we can also understand, for example, if there are invasive species you were not expected in your sample. So how is this useful for blue carbon? So for blue carbon, many of the projects we worked on are about restoration and preserving the seagrass or the kelp forests, understanding that the restoration efforts are actually working, the health of these ecosystems. And so you can track all that with eDNA, and also you can track the invasive species. But I wanted to talk to you about, well, sorry, this is an example of different coastal ecosystem projects that the company has been involved in. And it comes to my attention, probably, this is one of the biggest tracking recovery for kelp ecosystems. And this is in the UK, and it's sponsored by the University of Sussex. All of them have different QR if you wanted to get more information from them. Let me know if they work. Another very important project is the Kelp Forest Foundation. Kelp Blue rings a bell to you. They're also trying to establish blue carbon procedures and understanding how they will monitor this to get the additionality required for the markets. And of course, the Falklands or Malvinas, whatever you want to call them, are also a very important source of kelp forests. So these are the most significant examples on how DNA is helping to preserve and understand blue carbon. In particular, the company has established, I have this here, thank you. The company has established a methodology, and it's peer reviewed and published. Although Hannah was talking about this, and I was talking to her a little bit before this conversation, these two studies point at the precedence, the provenance, the source of blue carbon as something that eDNA is very good at identifying. So I invite you to go and read these papers. I'm just the messenger here. I'm not the specialist on this. But yes, so the organic carbon apparently is easy. It could be easier to add DNA to the source and the soup of different methodologies to understand where is this carbon coming from. The high-level conclusion of these two papers point at the eDNA processes and metabar coding as a proxy of these carbon sources. And they do claim it's the first study that taxonomically goes to species level, and it's able to discriminate between mangrove, seagrass, and macroalgae. The interesting thing, if this was true, that we can incorporate eDNA to the traditional isotope studies, is that it is very costly effective the way it is done. An example of what do you do once you collected the eDNA and you have to talk to different shareholders, stakeholders that are interested in making decisions is what this means to me and how is this helping me. So my job during the time I served at the company was to create insights that were useful. In particular, I'm going to show you something we worked on, which is connecting this eDNA into maps and telling the stories and understanding exactly how we can help decision makers. And the way you do that is to put this into something called species distribution models. And I want to ask you, who's familiar? Can you raise your hands, the ones familiar to this concept here? Tobias, too, okay. So I'll explain it. Species distribution models are statistical representations of where species could be found. This is basically an extrapolation or interpolation in reality of understanding once you have a species, you can also correlate that with environmental variables such as elevation, temperature, humidity. So the theory says that if you have shown a machine learning model enough samples of a certain species with certain characteristics such as the land cover and the elevation and the how much sun there is in certain area, etc., it should be able to pick up a new area that has never seen and tell you which species you should be able to find, right? So that's a species distribution model. And how is this useful? Once you have a species distribution model, you have statistical probability of finding the species or not. Let's assume you will trust this because we give you some confidence levels, and then we can inform where the species will be roaming around. And this is important for conservation and for restoration. On top of that, you can prioritize different species like red list species, you can prioritize invasive species, you can understand where they should be found and where should be restored. So for example, this is a mine here, and you can see different distributions, but the most important one is a distribution that we call the beta irreplaceability, which is the most difficult species. If we mess up with these ones, there will be big consequences. So that's the area where the species are. We should not go there. We should protect that. We should restore that area, basically. And so this is the kind of thing you can do a combination of eDNA and geospatial data. Okay, so that's good. What comes next is that there is a disruption in this space. Are you all familiar with ChatGPT? Yeah, well, this is the new thing. This is a ChatGPT for images. DeepMind from Google has launched 2 weeks ago Alpha Earth, which is a geo foundation model. It's a ChatGPT for Earth. It's not the first one. There's one called Prithvi that probably you know already. There is one called Clay, which is open source. And there will be many others in the market, but DeepMind is Google, and Google has been collecting data for the last 20 years or so, plus they incorporated all the open source data sets that are available. They trained that, and they made very efficient embeddings. And I'm going to go and talk about this. So what is this? The idea is that you can now interact with this compact amount of knowledge and ask very complex questions that will take a GIS expert weeks of manual work or labeling or training. For example, you can now ask, where are the swimming pools in the planet? And it'll pick up that because it was trained and now it can identify, and it can tell you, there are the swimming pools, there are 5 billion swimming pools. You can see the distribution here or there. That's not an important question from this conference, but imagine. You could ask what kinds of trees or where are the mangroves and how they are changing in time. And you should be able to pick up that quite easily. This is what they claim. So the embeddings, you don't need to actually go into this rabbit hole, but I thought I'll talk about this because I think this is the new way of understanding complexity. And I think biodiversity is probably a very complex problem that as an astrophysicist, I tell you, we don't have the math for this, but maybe the embeddings will tell things that no other system can actually crystallize. So what's going on here is that for a pixel, for a particular pixel, they aggregate all the layers of all the possible data, and they combine that also with other pointers of data, for example, infrastructure, species per pixel, water per pixel, and collect all that and let the convolutional neural networks that run through this pick up a new representation, which is small, is a vector, and that very compact thing has a lot of information. This is a new space of AI, and there's a lot of research that needs to be invested in this to understand what the embeddings are saying, but let me tell you a story about this. So DeepMind published the paper, and then they talked about this openly. They saw that the data they used to hear, this is bioacoustics, hear the birds, train the machine learning models, all the data from all these different birds talking to each other, chirping and singing, will create an embedding like this. So now they can identify any bird because it's pre-trained. Then they did the same with coral reefs. Again, listening to coral reefs. And what they discover is that the embeddings from the birds are very similar from the embeddings of the coral reefs, which is not a surprise if you think about that. That's just nature optimizing the frequency of sound in two different environments. But it wasn't so obvious in a crystal clear, transparent mathematical representation before these elements appear. And I think with geospatial data, we'll see the same. We'll see patterns of things that all these different labels of data will not give us an understanding humanly because it's too complex, but the embeddings could say, oh, there are patterns. I can identify an ecosystem because it looks like the same. And we don't have to manually target or label this anymore. So that's the rant. I wanted to say that in the machine learning world, having a foundational model is important because it's cheaper, it's easier, we can train things quicker. It's important for this field because we can now have seen change detection. We can understand comparing different landscapes very quickly. So for example, deforestation or wildfires, or if we wanted to see how the mangroves grow or retract, we could also see this. Similarity search and clustering are all very easy to do because the data is pre-trained, as I said. So my invitation is that it's open there in Google Earth Engine. Go and try and see, especially for the LiDAR community, go and try and see what it is doable, because I think it's going to be disrupted and it's just started. And to conclude, I'll say that if we can have ground truth and if we can combine the methodology of eDNA and these geospatial models into very comprehensive and coherent maps, we can accelerate overall what we are looking for, which is having a systematic way to account for what's going on and be able to report it and verify it and make these carbon markets more attractive for the financial flows of money. Yeah. So that's a message. Thank you very much.
Agradecemos a la doctora Jiménez por esa
Thank
interesante,
Dr. Jimenez
realmente
for that
interesante exposición.
very
Ahora
interesting
seguimos con
presentation.
el
Now we
doctor Jorge
have Dr.
Hoyos-Santillan, quien es
Jorge
investigador
Hoyos-Santillan,
senior
senior
en la Universidad
researcher
del
at the
Este de
University of Eastern
Finlandia y
Finland
el Instituto
and
Smithsonian
the
de
Smithsonian
Investigaciones
Tropical
Tropicales
Research
en Panamá.
Institute, Panama.
El doctor Hoyos-Santillan
Dr.
ha trabajado
Hoyos-Santillan has worked
extensamente en
extensively
flujos de gases de efecto
on greenhouse
invernadero y
gas fluxes
almacenamiento
and
de
carbon
carbono en
storage in
humedales
tropical
tropicales,
wetlands,
con un enfoque
with
en
a
integrar
focus
la
on
ciencia
integrating
en
science
estrategias
into
climáticas
national climate
nacionales. Su
strategies.
investigación
His
en
research in
Panamá
Panama
conecta con el
connects
monitoreo de
field-based
ecosistemas de
ecosystem
campo con
monitoring with
esfuerzos de
policy-driven conservation
impulsados por
efforts.
políticas. Hoy nos
Today, he will
presentará los
present
manglares en la estrategia
mangroves in
climática de Panamá. Le
Panama's
damos la
climate
bienvenida.
strategy. Let's welcome Dr. Hoyos.
Eso es email.
Aquí para volver.
Ok. Hola, ¿qué tal? Buenas tardes. Algunos de ustedes ya vieron esta
Some
presentación.
of you have already seen this presentation,
Entonces,
so
quizás se van a dar cuenta que
maybe
hay
you're
varias
going to
diapositivas que
realize
están un
that
poco
there's
recicladas.
several recycled
Iba a dar la presentación en
slides.
inglés,
I was
pero
going to do this
considerando
in
que
English,
estamos
but
acá
considering
en
that
Colombia,
we're
vale
here
la
in
pena
Colombia,
darla
it's worth
español.
doing it in Spanish.
De todas maneras,
Anyhow, any
en inglés, la
questions
pueden
that
hacer
you may
y
have in
quizá
English,
algunas
just
como
go
las
ahead
repito.
and maybe, some things I'm going to be
Les voy a
repeating.
platicar un poco acerca
I'm
de las
gonna
experiencias que
talk
hemos
about the
tenido en el caso
experiences we have
de
had in the
la
case
contribución que hemos
of
hecho
the
desde el punto de vista
contribution we've made
científico
from
a la
the
aplicación
scientific
de
standpoint to the
la ciencia
application
o
of
cuál es el rol que tiene la
science or
ciencia
the
en
role
la
of
implementación de
science
estrategias
in
climáticas
the
específicamente en Panamá.
implementation of
Porque
climate
la
strategies in
ciencia no
Panama,
es una
because science
herramienta
is
para
not
las
a
estrategias
tool for
climáticas,
climate
la ciencia es el
strategies.
motor
Science is
que de
the
hecho conduce la
engine
estrategia
that
climática de los
drives
países.
the
Y voy a
climate
elaborar un
strategy
poco
in
en
the
eso.
countries, and that's where I'm going to elaborate.
Hemos estado platicando
We've
acerca
been
de cuáles son los
discussing
mecanismos a través
the
de que los
mechanisms
países
through
han
which
podido intentar
countries have been
implementar
able to
soluciones
implement
basadas en naturaleza, que vamos a ver qué
nature-based
son, y
solutions. We'll see what
específicamente
that
los
is,
manglares como
and
parte de su estrategia
specifically
climática
mangroves
para
as part of
combatir el
their
cambio
climate
climático.
strategy to combat climate
Entonces,
change.
la
So
plataforma que
the
existe actualmente
platform
y
we
la que
have
acordaron todos los
nowadays and the
países
one that was
es
agreed
poner todas sus
among all the countries is to
propuestas
put
o
all
sus
their
compromisos
proposals or their
climáticos
climate
en
commitments
una estructura
into
centralizada que
a
se llaman
centralized
las
structure called
contribuciones determinadas
the
nacionalmente.
NDCs, the
Y en ellas lo que
nationally
hacen los países que dicen yo voy a
uh,
reducir o voy a
contributions,
mitigar o voy a
nationally agreed.
reforestar o voy a
Countries
conservar
say I'm
un
going
ecosistema
to
determinado, por
preserve,
ejemplo, en el caso de
restore
soluciones
a
basadas en naturaleza,
specific
o voy a disminuir el
ecosystem,
consumo de
or I'm
energía
going to
tanto, o voy a
reduce energy
cambiar
in
el
this
consumo de energía fósil y
matter,
voy a
I'm
pasar a
going
energía
to
eléctrica.
change
Y lo que va a
into
ocurrir es que entonces voy a
electricity instead of
sentar
fossil
un
fuel,
mapa o
and I'm
una
going
ruta
to
hacia volverme
lay
carbono neutral,
a
es decir,
path
la
to
cantidad de
becoming
carbono que está emitiendo actualmente un
neutral
país.
carbon.
Si
That is
tú
the
disminuyes
amount
tus
of
emisiones, llega un punto en que la
carbon. If
cantidad
you
que emites es la
reduce
misma cantidad que la que
the
capturas,
emissions,
entonces
is the
se
same
desbloquea.
amount that you
El caso de Panamá es
emit and
particularmente
capture.
distinto
In
y
the case of
especial porque
Panama, it's
Panamá
different because
se asume
Panama
como
is
carbono
assumed
negativo.
as
Es uno de los
negative,
pocos países en el mundo que se asume como carbono negativo, pero hay
carbon
un par de
negative. Some of the few countries in the
asteriscos que
world,
hay que
but there
poner
is a few
en el hecho de que se
caveats
asume
that
como
we
carbono negativo,
have to
como lo que
explain
definen
as
como bosque.
negative
Y eso es importante porque hay países, como el caso de
carbon.
Surinam, como el caso de
There are
Panamá, como el caso de
examples such
Finlandia,
as
o lo que estaba
Suriname,
considerando
Panama,
de Chile,
Finland, or
que gran
Chile,
parte de su
where a vast
cobertura
part of their coverage
territorial es
in their
bosque.
territory is
Entonces, de
forest.
hecho, el único
So this is why the
componente
only
a nivel
component at
nacional
the
que
national
es un
level
capturador
that
neto, un
is
secuestrador neto de carbono son los bosques en
a
muchos de estos
net
países. Panamá, por
carbon
ejemplo,
is forest. Panama, for
cerca del
example, is, I
60%
mean,
de su territorio lo que
60%
está
of its
cubierto de bosques.
territory is covered
Entonces,
by
el
forest.
único
So the
componente
only
de todos los componentes
component
de
of
sus
all the
NDC que de hecho
components of
contribuye a la
their
captura de carbono de
NDCs
manera neta.
contribute to
El caso de Chile es
their
el mismo.
carbon
El caso de Finlandia es lo
sequestration.
mismo.
Same thing in Chile, same
Entonces,
thing in
ellos
Finland.
tienen
So
un
they
compromiso
have
particular con las
a
soluciones basadas en naturaleza
special
porque les
commitment
interesa
with
mucho
nature-based
conservar sus
solutions because they're
bosques
very
y o
interested in
promover la captura a
preserving
través de
their
ellos.
forest and promote
Es importante mencionar que las
carbon
soluciones basadas en
sequestration. So
naturaleza son
nature-based
voluntarias, es
solutions are
decir, los países
voluntary.
las
That
asumen,
is,
pero
countries
son
assume
legalmente
them, but they're
vinculantes,
legally
se comprometen a
binding.
hacerlo y están
They're committed to
reguladas.
doing it, and
Asimismo,
they're regulated.
son
Likewise,
iterativas, es
they're
decir, que cada 5 años tienen que
iterative. That is,
revisarlas
that they have
y
to
eso
be
es
reviewed every
importante
5 years,
porque
and
hay un
that's
componente
important
político muy
because there is a
importante
very
de lo que
important
va a
political
depender después
component on
si los
which
compromisos que hicieron de manera
I mean,
voluntaria se van a
those
respetar o
voluntary
no.
commitments are going to
Y
be compared
finalmente,
against,
que todos
and
los
finally,
países
that all the
contribuyen de
countries
manera
contribute
similar, poniendo
similarly,
todas sus propuestas, al menos
putting
todos los que
all their
firmaron el
proposals, at
Acuerdo de
least
París,
all those that
para que
signed
de
the
manera
Paris
global
Agreement, so
combatamos
that
el
all
cambio
globally
climático.
can combat climate change.
Hace rato hicieron una pregunta acerca
You asked a
de cuántos de
question
los países
about
en
how many of the
Latinoamérica
countries in
tenían
LATAM
o habían incluido en las
had
soluciones basadas en
included
naturaleza
nature-based solutions
los manglares.
uh,
Y este es
uh,
el caso, este es
for
el listado.
mangroves.
Me
This is
puse a
the
revisar
list.
todas
I
las NDC
checked
de los países
all
de
the
Latinoamérica.
indices of
Hay países en
Latin
Latinoamérica, pero
American
también hay
countries. There are
territorios en
countries and
Latinoamérica, es decir,
territories in Latin America,
países
that
o
is, countries or
territorios que
territories
pertenecen, por
that
ejemplo, al Reino
belong, for
Unido, pero
example,
que
to the
están
United
dentro de
Kingdom,
América
but that
Latina.
are in
Y lo
Latin
que
America. And
encontré es
what
que
I
del
found is
total de
that out of a
territorios de
total of
países de América
territories
Latina,
and
únicamente
countries in LATAM,
16
only
tienen
16
incluidos los
have
manglares de
included
manera literal
mangroves,
en
so
sus NDC,
literally in their
que no quiere decir que no
NDCs.
estén,
It
quiere
doesn't
decir que
mean that
están de
they're
manera literal,
not
se
there, but they're
mencionan.
literally there, they're
Y no
mentioned.
solo eso, después,
And not
al ver
only
todas
that,
las
then I went
NDC,
to alert
lo que me
the
di cuenta es que
NDCs, and
únicamente
what I
7 de
found
esos
is that
16
only seven
son
out of those
compromisos
16
cuantitativos, porque las
are
NDC pueden ser
quantitative
cuantitativas o
commitment, because
cualitativas.
it could be
Con
qualitative or
cuantitativas me refiero
quantitative.
a que un país dice, yo
Quantitative
voy
is that a
a
country says that I'm
conservar
going to
1 millón de
preserve, I don't know, a
hectáreas,
million
como Chile, que
hectares,
dice,
such
yo
as
voy a
Chile.
restaurar 1
They
millón de
said I'm going to
hectáreas.
restore a
O puede
million hectares, or it can be qualitative, like Chile or Panama that said that we're going to map an ecosystem so they don't commit to an area or X quantity of carbon, but they just say they want to know something.
En el caso de Panamá,
In the case of Panama, for example, We're going to see that in a moment, but Panama went from qualitative commitments to quantitative commitments, and that's the natural jump that countries take. This is what we're doing. Once a country has enough information, then they can go ahead and do qualitative commitment, which are the ones that are worth from the economic perspective. So why nature-based solutions? As I said, countries come with their proposals. They say they're going to reduce their emissions, but they needed some sort of mechanism that was cost-effective. That is, that was cheap, and that gave them a high profit. And after several studies in 2017, they developed the concept of nature-based solutions. That is, the conservation, restoration of ecosystems for carbon sequestration that allows you to get carbon credits and sell those carbon credits in the world carbon markets. In the case of Panama in 2020, they published their first NDC, and in that NDC, they mentioned mangroves literally expressly, but as I said, it is qualitative. They say that they were going to integrate mangroves in their national carbon balance, so that they're going to write a manual for restoration of mangrove, but they don't establish specific number of tons of carbon sequestration. But it's different when you have a commitment, because this is legally binding. So once you are committed to doing something, you have to be able to monitor and quantify it, and this is what we're trying to do in this meeting. So then Panama had to update their NDCs, and what we did is that we had the possibility to have access and meet the Ministry of Environment and other stakeholders, but that is Ministry of Environment, IDB, and many local stakeholders, and we realized that it is true that Panama had the mangroves in the list. The message could be more direct and say it would be worth saying that they included these mangrove ecosystems in a quantitative or qualitative. It didn't have information, so we participated directly in writing some of the entities in Panama. This is the first step that I just mentioned. How can we integrate mangrove in the climate strategy of a country by committing to the system, saying that I'm going to do this for the system, or at least recognizing Chile hadn't recognized the amount of mangrove they had. And it was until 2019 that we were able to publish a paper saying that there's 3.2 million hectares. Then Chile said it was worth preserving them. And in this new update last year, they commit officially to increase the coverage of mangrove and also integrate them in the national system of protected areas. Interestingly, is that they have these quantitative commitments in the case of mangrove, but you're going to see that there are a lot of loopholes from the methodological point of view, from the information point of view. Simultaneously, I wrote a part of the commitments, and what I said is that they had to commit to map these ecosystems because they didn't know, and estimate carbon with this, which is not including any specific number. So this is the integration that they did. The detail we have now is that in order to integrate any ecosystem in your nature-based solutions or your NDCs, and to preserve or restore an ecosystem, you have to know where it is located. You need to be you know where it is and contextualize it. And those are all challenges that countries have. Going back quickly, in order to implement this, countries must have, I mean, you can see that, but they must have infrastructure, human resources, financial support, and they also need to have a political will. In many cases, countries have the HR, infrastructure, financial support, but they lack political will. So there's no integration in public policies. Therefore, there is no clear roadmap for implementation. The specific case of Panama, challenges were there. That is, they didn't know where mangrove was located because there is a map definition is pretty wide and have room for improvement. And there was other studies before, but these are limited studies with the specific areas in the country where information was not published or public. And in the case of greenhouse emission, there was no information. In case of the extension, what I can show you is that Panama, in fact, there was an estimate that since since 1969, it had lost more than half of its mangrove. Reality is that every estimations in coverage of mangrove in the countries, in the case in Panama, depend on the methodology used. That is, estimations in 1969 were aerial photos, and they were made not with so much detail compared with the high resolution we have nowadays. And more importantly, from year on year, technology changes, what Dr. was saying before. There's a new methodology with artificial intelligence, so you can have high def images where you can identify some of a specific species that can be present in terms of proximity. And then you use different resolutions. 2015 map had these 30 meters pictures, and 2022 is 10-meter images. So you have just different things. For carbon stock, variability and uncertainty is brutal. When I did the original estimations, it could go from 25 to 65 million tons carbon, and the area changes depending on the reference and what Hamish and Toby said. Nowadays, the allometric equations don't necessarily reflect or allow for adequate estimations. It all depends on the equations you use. That's what you get in this huge variation that could be 200, 300% can derive from the allometric equation you use for every species. Even the same species in these countries that I just commented, those allometric equations vary. Something else important, as Toby commented, is the estimation of carbon in soil. Different countries use different methodologies depending on the availability of their budgets, and what happens is that they use different depths, or like Hamish said, where you can show 1 meter, 2 meters, depending on how deep it can go. So the studies that report 20 centimeters, 30 centimeters, a meter, half a meter, and they don't publish the information raw, but they just publish the whole reservoir. So that takes us to another problem. You generate information, you have a human resource, you have a people, you have a funding, but you don't have the information for anyone else, just for you, and then it get lost in time. These were some videos, but I think that we won't be able to show them, but you can measure carbon dynamic in different components in soil, in trees, and then you can add layers on top of those different dimension components. That is, what's the impact of the fluctuation of the tides? Or Hamish said, we're going to estimate the superficial area of a mangrove to understand the tree, but there is some dynamics there. So the greatest part of emissions is in the base of a tree. So you can have a total area of a tree, but you can just multiply by one single number. And another component in the current situation of Panama is that there is a brutal uncertainty in relation to what's going to happen in the future. That is, we know we have some projections and forecasts in terms of the raise of water levels, but we don't know what's going to happen with carbon. In the case of human resources, when we conducted a study that we did during 3 years with my colleagues, what I found is that all the people that worked in the field with me in Panama, we were all working hand in hand, but the people doing all the models, estimations, and analysis, estimates, and they had to be all Panamanian. That is, we build capacities. We were able to generate. Joemi graduated with us, and the Victor wrote his thesis with us too. So what happened is that we had the HR, and now they have to become the PI scientist. If we want this to stay in time, it's got to be at the national level. You cannot just keep bringing people from abroad to do this research. This says that this is that the guys published that they actually developed in the case of infrastructure. In the case of Panama, we had to use the facilities of the Smithsonian. Here you have the INVEMAR, but their country, they don't have a Smithsonian or the INVEMAR, so it becomes impossible to conduct any research. Very easy things such as where to store samples, so that becomes a big win. And maybe these things look easy, but this is the difference between having a usable sample, and estimations can suffer there. The financial support, again, in the case of a project that I worked at, was the IDB, and it was executed through Audubon, but there's also other funds, the UK, the JEF, but for this to be sustainable in time, you need a national market or national funding that can support this along the years. If you continue to depend on small grants or pilots, Simply, you don't have the possibility to thrive in time, including monitoring, quantification, verification. And then I mentioned political will. There are countries that develop the roadmaps for the implementation of nature-based solutions. But at the same time, maybe there's some other projects, for example, mining projects, so it becomes mutually exclusive. You cannot approve mining in places or sites where they have forests because they're mutually excluding. So if there is a political will, it's got to be consistent in time. The projects such as the one that I worked at, or some projects like this that contributes to the implementation of climate strategies. My group in this project, we worked, we estimated reservoirs, baselines, we measure gases, and we estimated the total areas and carbon reservoirs in these ecosystems in Panama that represent 14% of the mangroves in the country. That's not the total. We also did estimates or maps at the national level on the coverage and carbon reservoirs, and we projected what was going to happen once these sites got flooded, given the rise of water level in the sea. This is some progress we've made. We have also found that we don't have numbers on restoration rate, because when you restore, you need to know where and why it failed or succeeded. And what we realized is that there was many projects that have failed, and they wanted to restore again in those sites that had failed. And this is going to be a constant in function of climate change. There are just sites that are not viable for restoration in 10 or 20 years. So maybe the most adequate strategy is not just trying to restore a specific place or site. There are projections for those restorable areas, but that are not applicable. Finally, in the case with Hamish, what we're doing is that there are some other uncertainty factors, which is the equations that I commented before. And then you can go to a field, we're doing all this, working with the trees. And in the case of the keys in Panama, Alicia, Javier, Kevin were able to create the maps. This was animations, but they just didn't come through with the presentation. Finally, information that I was referring to, information that has to be generated, has to be open or be published, either by governmental stakeholders, that is directly by the ministries of environment of the countries, but also it's got to get down to the local communities for them to know what value they can get from their ecosystems. There are cases of companies that arrive in countries, for example, in Panama that sell carbon credits. People are not fully sure what they're buying or selling, and then they give them the money, then they cannot do anything in the land for 25 years. So there is conflict of permanence of the projects and tenure of the land. It's important that the national, local academy or academia involves in get involved in these projects, and because they need to survive in time, and scientific community has to be has to have access to all information. We publish information in scientific data for everybody to have access in the above ground biomass. This is all the trees and everything we've done. It is being reviewed, the soil portion of it, but I also included all information we have on soil groups in Panama and the one that I have, and that's going to be a fundamental tool that can be used for comparison in the region for the project as a whole. And altogether, this is going to allow that climate strategies can be implemented and inclusion of mangroves in these countries. Otherwise, like in the case in Panama, there's still challenges to be overcome.
Muchas gracias, Dr. Hoyos.
Thank you very much, Dr. Hoyos. Up next, we're going to have the presentation of Dr. Jasper Feyen, and he's a researcher with a doctorate in the Ghent University, Belgium. Dr. Feyen is specialized in applications of teledetection for monitoring of mangroves, combined with LiDAR, drone images, and also satellites to create structural profiles of high resolution of the coast forest. And the work in Suriname helps to establish a robust baseline for the accountability of carbon, the evaluation of the health of these ecosystems. He's going to present the characterization of mangrove structures in Suriname using terrestrial LiDAR and multi-source remote sensing. So we give you a warm welcome.
Yeah, so hello everyone. So my name is Jasper. First, a small correction. I'm not a doctor yet, so I hope to be in 2 years or something. So I'm just doing my PhD for now. So indeed, I will give my talk today on the characterization of mangrove structure in Suriname using multi-source remote sensing data. So we did a lot of data collection in the mangroves with multiple tools of TLS, also aerial, ULS, so UAV, LiDAR data, and also with JEDI data. So what I want to do is, first present myself a little bit, like who am I? Then I will continue with talking a bit about the mangrove mapping that we have done within the area. Then I will also show a little bit of our terrestrial laser scanning data. Then on a study that we did with JEDI data, again, structural assessment. And as last one, I want to talk about a small pilot study that we did along 2 mangrove chronosequences with a UAV LiDAR. So first of all, who am I? It's quite a philosophical question sometimes, but this time I will keep it technical. So in 2018, I started working at the SBB, which is the Forestry Institution of Suriname, where we started working on a mangrove project, a mangrove inventory project of the GCCA Plus project. We first set up about, at that time, it was 12 sampling units in the mangroves of Suriname. That we continued in 2020 and 2023 with a second GCCA Plus project where we remeasured the plots and also established new sampling units within the area. So now we have about 17 sampling inventory plots. But I will not talk a lot about that because my colleague Valentin of SBB will also give a talk on that in, I think tomorrow it will be, so on a national scale. So I will focus on the research question of my, my own PhD. So first of all, let's go to the mangrove mapping. So yeah, we want to know where the mangroves are, of course. So first we looked at, can we map the mangroves dominating species with Sentinel data and using Google Earth Engine. So we looked at 3 mangrove classes, the red mangrove, Rhizophora, the Avicennia, the black coastal ones. And we also considered a 3rd class that gives us the mangrove graveyards or decaying mangroves, because that's also a quite extensive area in Suriname. So we did this with Google Earth Engine with just a simple random forest model. So I definitely want to test that new system as well and see what it does. Secondly, we also looked at the historical cover in Suriname using Landsat data, again in Google Earth Engine, and retrieved a yearly cover map of the mangroves. So we could get yearly cover estimates, but also based on the time series, we could reach age maps of the mangroves up to, what is it, about 36 years old, and also see the mangrove changes. So during our second GCA inventory phase, we also brought a TLS scanner to the field. So it was already mentioned by Toby in his presentation. So we took it in to a total of about 20 plots, where we measured about, I think 20 different types of mangroves. We tried to spread them along the different structural types, from juvenile ones, so the young mangroves, till the older ones, declining ones, even the graveyard stands of the black mangrove. And then we also checked or get some plots of the Rhizophora one, so the dominating red mangrove, and some mixed plots with both species in it. So in the next 3 slides, I just want to show some imagery of that, because not only is TLS data very useful, but it's also very cool to look at. So normally this should be a video. Unfortunately, it doesn't work. Too bad. Does it work? Let's hope. There we go. Yes. So this is a first plot in a mixed type of mangrove. So you can clearly see the roots of the red mangrove, but in between you also have the black mangrove. Yes. Okay, maybe we can go to the next one. So the next one is more at the eastern coastline, which is a young Avicennia, so black mangrove, like estimated with, again, the Lancet data to be about 14 years old. So very dense, a lot of trees stacked together. So this we have as well. And then the last one was probably the most difficult one to get to with the scanner. It was a red mangrove forests within fresh water. So we had to climb first 200 meters of aerial routes with the scanner to put the scanner in the water. And definitely don't know if I would do this again with this kind of equipment, expensive equipment, because it was really dangerous. But at least the data is very cool. Okay, next I want to talk about a study for which we submitted a paper about 4 months ago and is still in review. So we looked at how JEDI data could also give us an idea of the mangrove structure in Suriname. So for those who are not familiar with JEDI, JEDI is a spaceborne type of LiDAR attached to the International Space Station. So it also has LiDAR shots, but compared with TLS, it's very broad scale because it's like a shot of 25 meters in diameter. So you only get like an idea of the vertical distribution of the vegetation in your forest. So you get these kind of waveforms, and you can see like on which heights is the most vegetation distributed. So what we in total we used about 9,400 high quality shots that were taken between 2019 and 2023 in Suriname. So quite a lot. What did we do? We first labeled the shots with the age that we got from these age maps from the Lancet data. Also, we checked what the distance was till the shoreline, because this distance to the shoreline is mostly a proxy of the salinity and the sediment depositions and the tidal influence and so on. And then we connected it with with metrics coming from these JEDI waveforms. So basically, we tried to make some models to see like what is the influence of both the age and the seaward distance on this JEDI structural metrics. So what were the highlights of this? That both have a distinct impact. So they are highly correlated as well, but they still have a separate impact on structure. So if you look at these called partial dependence plots of our model, if you look at the age, we see this typical exponential growth of mangrove the first 10 to 15 years, for example, in this metric, which is the relative height at 89%, which is basically the canopy height. And if you look at the influence of the coastal distance, that we have a more linearly transect up to 2 kilometers and a half. And then we get from that distance in general, like a transition to the more decaying or open swamp forest. So the thing is for the JEDI data is that a lot of these data coming from it, like the biomass density, are still models themselves. So they still need ground truth data and calibration. And for that, we can again look at LiDAR, terrestrial laser scanning data, or aerial or UAV laser scanning, which brings me to my next small research that we did do last year in 2024, again, together with SBB and also with CELOS, which is also the Centrum for Agricultural Research in Suriname. So in this study, we looked at two transects that go along these different ages. So from the young at coastline till the older and even decaying mangrove plots at the end of the mangrove forest. Same for the second transect, from the young to the old ones. So we established plots of a diameter of 25 meters, did some measurements in it, with the goal to see how, again, mangrove is changing from the young ones at the seaside till the end of the mangrove. So this successional development. So what we took the UAV laser scanning data, forest inventory data, took also soil sample along this transect, which is now part of the thesis of Raisa, which is a thesis student in Suriname. And lastly, we also took wood core samples, which was the subject of a thesis student of a student of mine in Ghent. So with these wood core samples, we used X-ray chromatography to get the wood density and also get some wood characteristics along this transect. So then we also flew it with a DJI Zenuse LiDAR drone, the whole transect, which also gives us some structural metrics from the young ones till the old ones. And we can also clearly see like the density from a high density plot with about 3,300 trees per hectare till, if you go further in the forests, then 1,600 trees per hectare. So this, how do you call it, this automatic self-thinning thing that's going on. And the second thing, I also tried to see if we could like separate each tree from these Zenuse data, and that seems to work quite well. So there's some research on this as well, like which tree parameters can we get from this ULIS data as well, which is of course not as detailed as the TLS data. So lastly, what we want to do with this as well is to use this ULS data to simulate these JEDI shots to link our field measurements actually to JEDI and to do this calibration. So in the end, we could like have improved mangrove-specific JEDI models, for example, for biomass again, but also like see if we can map mangrove density, loris height, or all these canopy metrics as well with JEDI and with some models that we are making. And finally, if we have these calibrated JEDI data, we're also looking into combining it with other remote sensing data, like Sentinel satellite data, or hopefully the new biomass P-band radar that has been launched somewhere this year. So see if we can use the JEDI to train the Sentinel biomass stack to get also again improved biomass and structural mangrove maps in Suriname. So that was it. Thank you for the attention. If you have questions.
Bueno, según la agenda, teníamos programado
Okay,
para
now
este
following the agenda, we had a coffee break program for the moment, but for our afternoon to be more productive, we're going to go ahead directly towards the panel. So then I invite the 4 speakers to come back to stage for us to do this panel now.
Por favor,
Please come to the stage.
pueden acercar nuevamente.